Okay, so welcome to this European webinar series on microglia. I'm Guy Brown from the University of Cambridge, and my co-host today is Tim Burkle. The series consists of monthly talks of 45 minutes by European scientists talking about their research on microglia, followed by a question and answer session. And today's webinar is by Jochen Herms from the DZNE in Munich, and he's going to be talking about microglia and its fundamental roles in synaptic pathology in Alzheimer's disease. So during the talk, please write any questions that you want to ask in the Q&A session in the chat function as the speaker speaks, rather than leaving it all to the end. But direct those questions to all in the chat function of the Zoom. But please keep your microphones off now, muted, and cameras off to preserve bandwidth. So I'll hand over now to Tim, who's going to introduce the speaker.
Great. So yeah, welcome everyone. Lovely to have you with us, and Jochen as well, of course. So to introduce Dr. Herms, by applying morphological, biochemical and electrophysiological methods and modern imaging, the lab of Dr. Herms is best known for its work on synaptic dysfunction in neurodegenerative diseases. Basic studies have been conducted on the physiological synaptic function of proteins critically involved in these diseases, particularly the prion protein, tau, synuclein, presenilins, and amyloid. As one of the first labs in Europe on the topic, he established long-term in vivo two-photon imaging in mouse models of neurodegenerative diseases to understand the kinetics of spine loss throughout the progression of neurodegenerative diseases. He was also the first to show that nerve cell and synaptic loss in an Alzheimer's disease mouse model depended on microglial function. He's currently the head of department at neuropathology at the LMU in Munich, and runs a research lab, the DZNE. So welcome, Jochen.
Tim, thank you so much for the kind introduction. It's really a pleasure for me to talk to you because I work on synaptic pathology in Alzheimer's disease now for 25 years, and I would like to share my ideas about this topic with you. So I'm working at the Ludwig-Maximilians University in Munich, and it's actually the university where the disease was first described. Here you see Alice Alzheimer sitting on a boat on Lake Starnberg on the left, and on the right you see Emil Kraepelin, the head of Alzheimer at that time of the Department of Psychiatry. And in 1910, Kraepelin wrote a book, textbook, and within the textbook he named a disease, Alzheimer's disease, based on a case description of Alice Alzheimer's. Alzheimer published a few years earlier in 1907 über a strange disease of the cortex. This was a description of a patient Auguste Deter Alzheimer saw in Frankfurt when he was working there as a doctor. And then he moved to Munich, and the brain was given to him years later. And so you see, we still have the section in my departments of this first case. And you see written on the object holder the town where the patient was from and the name of the patient. Nowadays, of course, you won't write names on object holders. Auguste Deter died with the age of 55. And actually, even though Alzheimer is smiling on the photo, he died a few years later on bronchopneumonia. So at that time, 100 years ago, people didn't become old. And that was the reason why this disease, Alzheimer's disease, was not common. And by chance, Alzheimer saw a patient with a familiar form of Alzheimer's disease, which is most likely a presenilin-1 mutation. So if you look at this section from Alzheimer, this is he played around with silver impregnation. So what he mainly observed was these intercellular black things. So what we now name neurofibrillary tangles within neuronal stomata, and we now know this as the protein tau that is aggregating there. And in addition, he saw all these small things, which are so-called neuropil threads, which also consist out of phospho-tau within axonal dendrites, and then the senile plaques, which are very. He saw these thick swollen axons in the middle of the dystrophic aggregates here. The protein that is responsible for these plaques, the beta amyloid, of course, he did not see. What is tau? Tau is a protein that binds to microtubuli. It's primarily in the axon where it holds the microtubuli together. And in the disease situation, it gets hyperphosphorylated and detaches from the microtubule and then aggregates in form of these neurofibrillary tangles. And these tangles is what we detect with antibodies for diagnostic purposes. And based on the elegant work from Michael Goeder's lab, we know now the structure of the tau fibers. There are two, the 4-repeat and the 3-repeat tau that is aggregating in Alzheimer's disease. So the tau pathology is the middle of the pathophysiology of Alzheimer's disease. It causes the neurodegeneration. It also causes glia degeneration, both astrocytes and oligodendrocytes. And these astrocytic pathology, tau pathology, is mainly due to the fact that astrocytes are involved in the lymphatic drainage of phospho-tau. The tau fibrils, the big tau fibrils are probably not most toxic. The most toxic tau species are probably oligomers or small fibrils, which are probably most toxic. During the last years, this neurodegeneration is accompanied by synapse loss. This is a prominent feature in human cases of Alzheimer's disease. And during the last years, it becomes clear that tau propagates throughout the brain in a similar fashion like prion protein. However, different to the prion protein, it takes much longer. It's about 10 to 15 years until the pathology starting from the temporal lobe will affect the entire cortex. So there's a prion protein-like propagation, which may be the cause of the so-called temporoparietal progression of tau pathology in Alzheimer's disease. So the tau pathology starts at the medial temporal lobe, and based on the stages from Bragg and Bragg, we stage this 1 and 2. This is without any clinical findings. However, mild cognitive changes occur if the pathology further progresses into the temporal cortex. And this is very easily detectable for a neuropathologist. So we usually analyze this part, which is marked here. We stain it for phospho-tau, and you even do not need a microscope to see these strong tau pathology within the temporal lobe. So the dementia then comes when the tau pathology further progresses into the cerebral cortex. So it's clear that the tau pathology strongly correlates with the clinical cause. So tau is critical for Alzheimer's disease. It's therefore that Alzheimer's disease is a tauopathy. However, Alzheimer's disease is not Alzheimer's disease if the A-beta pathology is lacking. So the A-beta pathology is critical. So A-beta has been described 40 years ago based on the analysis of vessels obtained from Down syndrome patients, where they find a peptide, and this amyloid peptide is what aggregates to these plaques. And the amyloid peptide is a part of the so-called amyloid precursor protein. It's a transmembrane protein that is cleaved by certain protein complexes, the gamma secretase and the beta secretase complex. Based on the cleavage of the APP, the A-beta peptide occurs, which further on, if it's not taken up by microglia, it aggregates into oligomers, and it further on aggregates to fibrils and plaques. So the A-beta peptide is critical for Alzheimer's disease. And that's indeed the beta amyloid that causes the tau pathology becomes clear if you look at the familiar cases. The familiar cases most often are due to mutations in one of the proteins of the gamma secretase complex, the presenilins. So mutation within this cleavage complex does cause a tremendous amyloid accumulation, and the development of tau pathology. So we have learned a lot from PET imaging of familiar cases with Alzheimer's disease. So PET imaging allows to detect the fibrillar A-beta aggregates in patients. So it's a radioactive labeled ligand of the A-beta, and here you see brains from a patient that has a mutation in presenilin over time, so 20 years prior to the disease onset and at disease onset, and compared to a non-carrier. And it becomes obvious that long before the patient saw any symptoms, so at 0, they have the first symptoms, the A-beta is accumulating in the brain. And this paper by Bethmann, it was quantified, and as you can see, the A-beta deposition in the precuneus starts 20 years before the start of the clinical symptoms. So the brain is more or less full of A-beta already 10 years prior to the disease onset, even though the patient don't have any cognitive signs. So A-beta accumulation in the brain does not cause dementia. However, pathology, like tau, has a prion protein-like propagation. It takes longer than the tau propagation, or of course much longer than the propagation of the prion protein. And somehow it causes the tau aggregates, which also spread throughout the brain. And the tau pathologen is the cause of the neuroglia degeneration of the synapse loss. There's another hypothesis around for 20 years now, claiming that there are toxic A-beta oligomer species that affect neurons and synapse independent of the formation of tau. From my point of view, this is still a hypothesis. We don't know whether these toxic A-beta species are really in the alive human brain. And I have problems in believing this because if you go back to this findings here, so the brain can develop lots of amyloid plaques without any kind of cognitive change. So the toxic effect of A-beta oligomers in vitro, I have difficulties to believe that this is really something that occurs in the human brain. Okay, but going back to the A-beta pathology, for sure they induce the tau aggregates. The mechanism is not understood so far. What we know is that this is only one way, because there are familiar dementia cases that cause tauopathy, but those patients that have this kind of tauopathy with dementia don't have A-beta pathology. So for sure, the tau pathology does not induce the A-beta pathology. The next question is obviously, or is the question, is this exclusive? So does A-beta pathology only induce tau pathology? This is actually not the case. About more than 50% of Alzheimer cases do have alpha-synuclein pathology. You know, alpha-synuclein is the protein that aggregates in dementia with Lewy bodies or in Parkinson's disease. So it's for sure not exclusive, the induction of the tau pathology. You also have alpha-synuclein pathology in Alzheimer cases. And the last thing I would like to talk about is A-beta pathology always inducing tau pathology. And here we learned a lot from one unusual case. So this is a woman with a presenilin 1 mutation, which usually causes dementia at the age of 40 or so. But this person was cognitive normal until the age of 72. And the brain, and the PET imaging showed the brain is full of amyloid, but the patient don't have dementia. And a few years later, she died on malignant melanoma. And the neuropathology reveals that there's very little actually tau pathology. So A-beta does not always induce tau pathology. And the cause of that, I will show you later, why this woman did not develop severe tau pathology. So, and another point why that's problematic to think about A-beta pathology, it does induce tau pathology. If you go in the standard, let's say, sporadic Alzheimer case, you have this progression, as I showed you. And if you look into, I would like to show you a typical case of a Bracken-Park stage 4. You know, this is the stage where you have mild cognitive changes. How does tau pathology coincides with A-beta pathology in this stage? So if you stain for phospho-tau, you have this typical appearance of something brown. You see the entire tissue is brown. You have some neuronal inclusions, but you also, and you have this enhanced signal here, but you see throughout the tissue, there's a delicate staining of axon and dendrites. And this coincides with the mild cognitive changes the patients have at this stage. So if you stain for A-beta in this region of the brain, often you see very little plaques. And it's difficult to understand why you have this tau pathology. The tau pathology at the plaque is due to the plaque. So this, we said this dystrophic axons is the consequence of the fibrillar A-beta. However, we have difficulties to explain phospho-tau in neurites distant from the A-beta plaques. And this is the pathology that mainly contribute to synapse loss in this part of the brain. And here we have also an important issue with the mouse models. So if you look to a mouse model of Alzheimer's disease, it's actually an amyloid model. So it has a mutation in APP and PSNL1 and produces tons of amyloid, and which is shown on the left. And if you stain for phospho-tau on these kind of mice, you don't get this neuritic staining. The only thing you get is a slight phospho-tau positivity at the location of the plaque. So the dystrophic axons you get labeled with phospho-tau, but distance to plaques, you don't have any phospho-tau. So it brings me to the point, phospho-tau pathology distant to beta plaques is not detectable in an A-beta amyloid mouse model. So actually in the mouse model, and the most often used amyloid mouse models, we don't have this important A-beta pathology-induced tau pathology. So actually we are lost in translation at this point. The A-beta amyloid models are bad models for the disease we name Alzheimer's disease in humans. So, however, what is the cause of the A-beta pathology inducing tau aggregates, and how does the tau aggregates propagate throughout the brain, and how does it cause synaptic loss? And at this stage, I would like to introduce microglia, and microglia may fill these important gap in understanding the pathophysiology of Alzheimer's disease. So what is the evidence that microglia plays such an important role? I came into this business 15 years ago when Martin Fuhrmann established long-term in vivo photon in my lab. And Tobias Bittner was the first who looked at amyloid mouse models. And then the Sunny boys stained everything what you can stain off in order to follow it over time. So they crossed and the so-called 3-point TG AD mouse. So it's a mouse that expresses PS1 with the mutation, APP with the mutation, as well as tau actually. So it's not an amyloid model only. And they crossed it with TXTCR1 GFP, so to label the microglia. And then they crossed it also with a mouse that labels neurons, a subset of neurons. So, and in addition, they injected fluorescent dextran. So you could, with this, they could analyze microglia movement, the fate of neurons, and their location due to the labeling of the vessels. And so they, with long-term in vivo photon imaging, you can follow the same cells over time. And this is here 2 images of the same area with a time delay of 7 days. And you may see that the number of microglia at this one neuron is increasing. And another 7 days later, you see that this one neuron that was at the location where the microglia came together is lost. So there is obviously a microglia recruitment prior to the nerve loss. At this time, I was quite amazed that this. I would have expected the microglia comes when the neuron is dead, so it takes up the dead cell, but it actually starts before. We could, however, not publish this in Nature Neuroscience without the mechanism. So luckily, the GFP in the CXCCR1 GFP mouse is within the locus of the CXCCR1 receptor. So the homozygous mouse is lacking the CXCCR1. And we compared the nerve cell density and we looked at nerve cell loss in the mouse heterozygous compared to the homozygous CXCCR1 mouse. And as you can see here, the quantification reveals there's no nerve cell loss taking place in this AD mouse model if the CXCCR1 receptor is lacking. So somehow the communication between microglia and neurons is relevant for the nerve cell loss in this mouse model. So this was the first indication that microglia plays a role in nerve cell loss in an AD mouse model. At that time, I was really struggled by this finding that actually I didn't follow it up at that point. At that time, no one was really that convinced that microglia play an important role in Alzheimer's disease. So actually, I did the mistake also stopping it. However, during the following years, there was lots of genetic evidence coming up that microglia function may be relevant in Alzheimer's disease. So the way of getting an idea whether there's any genetic risk is genome-wide association study. And then those have identified more than 40 loci, and actually half of them relate to myeloid monocyte microglia function. That was really amazing. So microglia must play an important role. These are the genes that are involved in phagocytosis. And you can put them in various groups, so-called obstinence, phagocytic receptor and downstream molecules, cytoskeleton, endosomal network, and lipid cholesterol efflux. I would like throughout my talk only point to these so-called trace recognition proteins, which are already obviously very important, linking microglia function to Alzheimer's disease. So in the aging brain, oxidized lipids, debris, dysfunctional synapses, pathological protein aggregates like A-beta, they get recognized by obstinence. And the most important obstinants are ApoE and ApoJ, which are apolipoproteins, and C1q. So these are the obstinants, and they are actually released from microglia and astrocytes, and they bind to receptors on microglia. And the most important receptors are Sol1, TREM2, CD33, and CR1. And this receptor binding is thought to cause phagocytosis, or let's say engulfment at microglia. So the trash is actually taking up based on this protein cascade. So ApoE is actually probably the most important obstinant. It binds to Sol1 or to TREM2. And actually, as I guess you know, ApoE4 genotype is the most important AD risk factor. ApoE as an obstinant depends on the lipidation status, and ApoE2 has a high lipidation and binds more A-beta, so the uptake of A-beta is highly efficient in ApoE2. However, in ApoE4, there's low lipidation and a low phagocytic activity. So it's suggested that if you express ApoE4, the uptake of A-beta that is produced in the brain under physiological condition might be, the uptake of A-beta might be less efficient. So ApoE actually plays an important role at various pathologies. So it's involved in the fibrillar formation of A-beta. It's important for the propagation of tau, and it might also be important for the toxicity of tau. And I come back to this woman. Actually, the brain is full of A-beta and has no cognitive alterations. And the cause is this patient has a homozygous ApoE mutation. And this ApoE mutation probably affect the neuronal uptake of extracellular phospho-tau. So this patient really makes clear ApoE plays an important role for the toxicity of amyloid in terms of inducing tau pathology. So what do we know about the phagocytoreceptors on the AD risk gene list? So there's SOL1, TREM2, CD33, and CR1. SOL1 actually regulates phagocytes migration towards chemoattractants, which is very important in the microglia function. And TREM2 is critical for microglia recruitment to A beta plaques. So in order to get you an idea what that is practically meaning, some years ago, we looked into microglia recruitment to fibrillar A beta plaques. And we asked the question, is the recruitment of the microglia only taking place if fibrillar A beta is present? So we look into an AD mouse model, an amyloid mouse model, I must say, that forms plaques at a certain point, and we label again the microglia with GFP. This was actually done by Christine Nong at that time. So this is the same area imaged over several days, seen from -7 days up to 106 days. And as you can see, the microglia moves to the red fibrillar A beta plaque. So there's no change in microglia density prior to the occurrence of fibrillar A beta. And then you see also that there's an increase of the density of plaque. So this is higher magnification, the area of interest. You see that the microglia moves to the fibrillar A beta plaques and that the density of microglia is increasing at the plaque. So there's clearly microglia recruitment to fibrillar A beta plaques. And this is reduced in TREM2 knockout mouse, as shown in 2017. And it is reduced in Alzheimer patients who have a TREM2 mutation. So obviously, TREM2 is important for the recruitment of microglia to plaques. However, TREM2 may also be involved in general in the uptake of A beta, which is not shown in this kind of studies. So the last receptor I would like to talk about is the CR1 receptor, and the obstinance is C1q. This receptor, C1 receptor, does not only recognize C1q, but also C3 and C4. And there was an important paper showing that complement and C1q decoration of synapses mediate early synapse loss in Alzheimer's disease mouse model from Beth Stevens' lab. I mean, this is one of these, let's say, 50 to 100 Nature Science papers talking about Alzheimer's disease mouse model, but however, only working on amyloid mouse model without any tau pathology. So I would like to make this point clear. This is not an Alzheimer's mouse model. However, what they saw was a C1q staining, which was higher in the amyloid mouse models, say 20s. They found 100%, so 200% compared to wild type, stronger co-localization with the postsynaptic protein PSD95 with C1q. And they claim that this decoration of C1q causes synapse loss in these mouse model. I have certain problems with these stainings because, as you can see, the C1q staining is as dense as the PSD95, indicating that more or less all the synapses are labeled with C1q, which is difficult to believe that this is the eat me signal. However, if they knock out a protein involved in the cascade, they can rescue these synapse loss they see in this amyloid mouse model. There's also another paper showing the same thing in a tau mouse model. In the tau model, actually, in the PS19, there is synapse loss taking place. And here they show that there's also C1q decoration. I mean, what this paper show and the other one is that C1q decoration is involved in synapse uptake. So the idea is that altered synapses in beta amyloid mouse or in a phospho-tau mouse get decorated with C1q and activates the CR1 receptor and then induces phagocytosis of these altered synapses. So I think there are a lot of, I showed you some genes where it's very obvious that it plays an important role in Alzheimer's disease, but I won't talk about all the other genes. You will find this in this paper, which I have cited here. So is there epidemiological evidence linking microglia function to an enhanced AD risk? And indeed, there is. Microglia get activated under various conditions: obesity, reduced physical activity, little sleep or stress. These are all conditions that affect the microglia function. And chronic diazepam intake has also been linked to affect the AD risk via microglia activation. So I would like to talk about the chronic diazepam intake as one example of an epidemiological evidence link between microglia and AD risk. And I would like to present a story that was performed by Yuan Shi, Maureen Chow, and Mario Dorska in my lab, and asking the question whether chronic diazepam intake affect dendritic spine plasticity via microglia. So again, I'm talking about in vivo photon imaging. So a glass window is implanted into the skull of a mouse. It is often a transgenic mouse where certain proteins are labeled. In this case, it's a YFP mouse where subset neurons are green. And this allows us to follow the fate of individual dendritic spines over time. So you see this film below. This is the change in spine plasticity over more than a month. And yes, you see there's a small change. Some spines come and go. And this is the so-called synaptic plasticity, which is the fundamental basis of learning and memory. And this plasticity is depending on microglia function, which I will show you now. So in order to get an idea whether diazepam impairs synapses, we implant a glass window in the wild-type mice. And then we imaged every 5 days prior to the application of diazepam, throughout and afterwards, and to look whether the diazepam dose, which is comparable to a human dose, do something to spine plasticity. So this is the image we get usually. So this is one dendrite in vehicle and one dendrite in the diazepam condition over time. Of course, looking at it, you need to do the quantification, and the quantification reveals that the spine density, which is stable in the vehicle group over time, does drop with the application of diazepam over a week on a daily basis. So there's about 10% reduction of spine density with the application of diazepam. So we believe that diazepam affects the function of microglia in synaptic pruning. So microglia stay in contact with dendritic spines. And this is an image from us. I mean, here you see in blue the microglia, and in yellow a few dendrites with spines passing through the volume. You see all the microglia of this volume, and you see only a few dendrites. And as you can see, these microglia each microglia cover its own space where it screens the synapses within. And so this is in higher magnification. You see here one dendrite and one microglia, and you see that there's interaction. And we study this interaction by performing in vivo photon imaging. And I would like to give you one example, also that you get a feeling about the time scale of spine plasticity relative to the microglia action. And I think this is important because reviewer always ask for this kind of two-photon in vivo imaging, showing something to show the effect of the microglia, which is difficult. I will show you. So here is one dendrite now marked in magenta and a few spines. And here you have in green a microglia process that is getting into contact with these dendrite. And this is a sequence over 25 minutes. You see in the beginning, and over the first 15 minutes, there's the interaction of the microglia process with the dendrite, and then it's gone. So there's only a short time that the microglia looks at this one dendrite, and we followed this dendrite over several days. 3 days later, you see that the microglia process has a different location. It's the same microglia because it's within the domain of this one microglia, but the one process goes to a different place. Another 5 days later, it happens. What happens in terms of spine plasticity? You see that in the first image that there one spine shows up, and this spine is, however, not a stable spine because the microglia get into contact and eats it up. So this is a so-called transient spine, and the microglia is involved in the uptake of this spine. Another 2 days later, you see this is one spine that was present here is gone. Also, the microglia is gone. So the time scale of spine plasticity is very different to the plasticity of the microglia process, which is amazingly high. So microglia get into contact with spines and get into contact with spines which are dysfunctional and take it up. So you then can analyze what the microglia has eaten. You look in the stomach of the microglia, so in the lysosomes, and in the lysosomes you find the synaptic proteins. This is actually a very nice test to get to know what the microglia is eating and whether the synaptic uptake of microglia is enhanced. So we looked into the diazepam-treated animals, and indeed, the puncta of the PSD95 puncta within lysosomes is strongly increased if we treat the animal with diazepam. So microglia obviously have eaten synapses if you go for the chronic diazepam treatment. So we checked for the important role of microglia by killing by microglia depletion using a compound, PLX, which drops the number of microglia tremendously. So we treat the animal over time with the compound and then give diazepam. And indeed, under this condition, the diazepam, here's the control condition, does not drop the spine density anymore. So microglia activation is critically involved for the spine loss with diazepam treatment. So how is this mediated? So diazepam not only binds to GABA receptors, but binds to the TSPO receptor. That is a receptor on the mitochondrial membrane, especially of activated microglia, as well as astrocytes. So TSPO is an important protein that is upregulated with activation of microglia, and diazepam binds to it. You may have heard about TSPO before, because TSPO is a ligand for the most commonly used PET tracer for microglia activation. So TSPO is a prominent protein with microglia activation. Diazepam binds to it. So what we did, we looked at TSPO knockout mouse, and indeed, in the TSPO knockout mouse, even though microglia are present, if the receptor for diazepam, in this case TSPO, is lacking, we don't see spine loss taking place. So obviously, TSPO is very critically for the diazepam-induced spine loss. And also, we looked at the stomach of microglia. So we looked into lysosomes of animals lacking TSPO receptor. And here we do not see an increased amount of synaptic proteins. So synapses are not eaten up with chronic diazepam treatment if the TSPO receptor is lacking. So diazepam affects microglia function via the TSPO receptor and reduces spine density. So obviously, there's a toxic gain of microglia function that may explain the link of diazepam abuse with the risk of Alzheimer's disease. So the hypothesis is that the pathological overactivation of microglia may cause synapse loss in neurodegenerative diseases. And this may also not hold only true for diazepam, but also for reduced physical activity or for obesity. So all conditions that affect the phagocytic activity of microglia. So I would like to conclude that I hope I have convinced you that microglia may play an important role in explaining the important combination of A-beta pathology and tau aggregates in Alzheimer's disease, and that genetic and lifestyle factors that affect microglia function at synapses may be important for the risk of Alzheimer's disease with aging. So I would like to thank lots of people throughout the last 25 years working with me on synaptic pathology in Alzheimer's disease. And in case you would like to work on this in my lab, there are lots of open PhD positions. So thank you very much for your attention, and I'm happy to take questions.